Got it. Okay. Okay, so today we're going to discuss the SYN-B nuclease from wNo Wolbachia, sufficient for induction of cytoplasmic incompatibility in Drosophila. So just so everybody knows, reading something like this is like a really nuanced paper on CI, and so you're really in sort of like the thick murkiness of CI. So if it was a little bit difficult for you to understand, not as a CI expert, then hopefully I can help you with that. Okay, so let's see. So brief review. We discussed last time how when originally these CI factors, CIFs, were discovered, well, actually, they were discovered before, but originally when they were first characterized in Drosophila, there are these weird phenotypes where A plus B gave you CI, but B alone did not do anything, and A alone did not do anything. And so there was kind of like this question of like, what's going on here? And there were two different hypotheses. One hypothesis to explain this was the 2x1 model, where you need both of these to induce CI. And another alternative hypothesis was that this is just kind of a fluke, the reason that you're seeing this. It's still a toxin antidote model. And the reason, for example, you might have seen this in one of the studies was because in the one case, they could never make a B transgene insertion alone. And so the theory was it was killing the flies. And then if you add the A, that obviously makes sense that it would alleviate some of the toxicity. And then that could get you CI. Okay, so there's sort of like these two different hypotheses. And now we're starting to get into really like the murkiness of how might you actually figure out which of these is correct. Okay. And so the context of this paper is there's many different Wolbachia strains. Okay. So remember there's like we've talked about, or maybe we haven't talked about other Wolbachia strains. Each Wolbachia strain starts with a W, has a name. Its name starts with a W, and it's named after its host. So W. pip, guess where that comes from? Culex pipiens, yeah. So this is a mosquito Wolbachia. W. mel comes from Wolbachia from Drosophila melanogaster. And in this paper, they're talking about this wNo. Okay, so I'll talk about that in a second. But the first sort of like idea for how you might start to resolve some of this mechanistic differences is start looking at how different CI operons function. Okay, and so W-pip CI operon called SID, W-pip has been studied. There's another operon called SYN W-pip, which has been studied. And there's another operon called SID W-mel, which has also been studied. And this was the one that really gave the evidence for 2x1, the SID W-mel system. Whereas these systems were more evidence for TA functionality, okay? And so the question is like, well, how do you resolve this? One thought was to start looking at mechanisms of a bunch of other CI operons, so operons from other Wolbachia. Okay, within that context, there are about 5 different strains of Wolbachia that have interesting CI phenotypes in Drosophila simulans. Okay, so this is another fruit fly Wolbachia, or another fruit fly, which is very convenient for research purposes because it's very easy to grow and test and do experiments in simulants. Okay. And within D simulans, there are, let's see if I can name them all. There is WHA, there is WRI, there is wNo, there is WMA, there's WMAU, which comes from Drosophila maritonia, a different species, which was put into Drosophila simulans. And then I think there's another one, W. Keeley. Okay, so there's essentially there's a whole bunch of different strains. And each of these have different CI factors, different mutations in their CI factors. And so the idea was, well, let's start to characterize some of these and see how they function. So to give some contextual history on this, these strains were particularly important because a while back in 1998, so this is when I was 10 years old, they discovered these interesting Wolbachia that have this interesting phenotype, which is that they are called mod minus rescue plus strains of Wolbachia. And what do you think that means? What's a mod minus rescue plus Wolbachia? So think about CI. Like in CI.
Like they don't.
Wait, what? No, I think you were about to say it right.
I was, but it doesn't make sense. I was going to say, like, it
Am I-
doesn't kill them, but it does rescue them.
Yeah, that does make sense. So why does that not make sense? So yes. So think about the original CI framework. Like, remember that in the original CI framework, there's two things that have to happen. There's like two events. There's like induction of CI, which causes the sperm to be sterile, and then that's how you get CI. And then in the other cross, there's a rescue event happening. And rescue is going to happen in the egg. So it's going to be controlled by some factor in the egg. But induction of CI happens in sperm. So it's going to be controlled by some factor that's in sperm. So how might you get a strain of Wolbachia, or what can you think about or describe that strain a little bit more, one that cannot induce CI but can rescue it? Like, what would the gene architecture maybe look like? So if these are the CI factors, and if we are correct that these are the genes that control CI, what might you hypothesize would be the structural organization of a mod minus rescue plus system? How would it might be different?
It would just have the CYP.
It might just have an A. So if the A is the antidote, it might just have this CYP-A. Or what else could it be?
The CYP-B could have, it could be negative B ubiquitinating and negative nuclease. Like it may not have the...
Might have like a mutation or something. Yeah, it could have a mutation. So essentially there would be like two scenarios under which you could get a mod minus rescue plus if this hypothesis is correct. One would be you just have a CYP-A gene. Like you just have a rescue factor and you don't have the induction factor. Although that's unlikely because these genes are always like genetically linked. So probably the better hypothesis for what you would see is you'd see something that looked like this. You'd see an operon that has a functional CYP-A, and then you'd see some kind of like a mutation in CYP-B, which causes it to become a pseudo gene. So just to make sure everybody knows, what's a pseudo gene? Pseudo gene is like it was a gene back in the day, but then it got a mutation and it died. That's a pseudo gene. And you can get a mutation that's like a frame shift that would then cause a premature stop codon, and then that would essentially just like kill the function of the gene. Okay, so if we were, yeah.
What would you consider a truncated gene, or is that just like its own thing?
Truncated gene, so like if it expressed like this little chunk,
Yeah.
it's most likely that's going to be a pseudogene because it's most likely that is probably going to be a dead protein, like not doing anything. But it depends, right? It depends. If you had stop codon out here, like maybe it would still be functional. Who knows? Like that's sort of like a hypothesis. But essentially my point is, if you have this phenotype of mod minus rescue plus, and if our hypothesis is correct, there must be something wrong with that B gene by default, right? Okay. And so one of these strains, one of these rescue-only strains, the genome was sequenced, okay? And I'm pretty sure it's these, and I hope that I'm not confusing MA with MAU. I think MA is from Madagascar. I think MAU is from Drosophila maritonia. But this genome was sequenced. That's this paper here from Brandon Cooper's lab. This genome was sequenced, and when they looked at the operon, it was a syn operon. So it has a nuclease, double nuclease domain in the B, okay? And there was a break. There was a break in the B. So the prediction came true when they sequenced this mod minus rescue plus genome, okay? So that was sort of like at least a good sign that, again, like if you sort of, there might be this nuance, but we're pretty sure that these are the genes that are causing CI. That's good evidence for it. How this fits in with wNo, so now let me talk about wNo and why wNo is special. There are some papers, I can't remember what they are, so if you're online and you're watching this, you can maybe write it in the chat, the paper that proves this. But the reason that they knew that this MAU was rescue only is it wouldn't induce CI, but it could rescue wNo. So when they would do a cross of wMAU, female, cross to wNo, male, this Wolbachia could rescue that. Okay, so essentially that syn operon, one sec, this syn operon, even though it's broken, that A can still rescue the operon from wNo. And then they sequenced the genome of wNo, and guess what the operon looked like? It was essentially like an exact perfect match of this syn operon. So it was a syn system, except it was intact. It didn't have this like weird like mutation. So that's what wNo is, and that's why wNo has kind of been like special, is there's been these genomes that were sort of like a gold mine for comparative genomics, and wNo was one of these like real special strains. And so there was this bioinformatic evidence that suggests that there was this interesting relationship, but nobody had yet characterized the function of the operon. Did you have a question?
I've thought about it further, so I'm good now.
Okay. So just so you know, like that's the context of this wNo. And things get really complicated when you do research on Wolbachia because there's all these like different strains and it can become like overwhelming. But if you're wondering like why are they studying wNo, that's why this is interesting. Other just quick like review points. When this paper, so the context under which this research was performed, there are some things that people agreed upon and some things that people did not agree upon. So let me just review those things. So there's broad agreement that the A's bind B's, okay? So they bind each other, okay? There's broad agreement that it's a genetically linked operon, okay? You always find A with a B. Okay, so here's where it gets complicated, okay? There is broad agreement that B is necessary for induction, okay? But there's disagreement that it's sufficient. Okay, so you see these terms a lot in science, necessary versus sufficient. Do we know that hopefully we know the difference between these? Like sufficient means like that's all you need. So if B was sufficient for induction, that's all you'd need, okay? If B is necessary for induction, it doesn't mean it's the only thing. There could be that there are other things. So everybody agreed that B is necessary for induction, but there was disagreement that it's sufficient for induction of CI. Okay. And everybody agreed that A is sufficient for rescue, which means that A is the only rescue factor. Okay. So these are what people are in agreement on, and this paper is really trying to resolve this. If I can highlight it, this point right here. Is B necessary or sufficient for induction? That's what it's trying to sort of like figure out. Okay, now let me ask you guys, why is this important? Like from what you read in the introduction or what you think, do you think this is important or no, this is dumb? Like, why do you think this is important? Maybe it's not. Maybe I made you read a bunk paper. Could, I might do that. So you're linking it to the vector control. So it's good that the mechanism of CI is sort of like at the heart of Wolbachia applications. And remember, there were there was population replacement and there was incompatible insect technique. And CI is really at the heart of that. So understanding the mechanism of CI would be really quite beneficial. But actually, like, let me push on that a little bit. As somebody who studies CI, can we elaborate that point a little bit more? Like, we don't really need to know the mechanism as long as it works. Like, who cares what the mechanism is? If it works, it works. Why? I'm playing devil's advocate. Why would you want to know the mechanism? Why would you give a shit?
It's good for just biology understanding.
It's good for biological understanding, but I feel like that's still like kind of like a weak point. Like,
if you're trying to use it, for other species, I guess. Possibly it would help you to know the mechanism, whether it's just the syn B or you need them both.
That's a really good point. So one thing that people have always thought about CI is it could be like a broad spectrum tool for all insects. And it would be helpful to know the mechanism of what it is to maybe see if that was true, or maybe that could provide insights into whether it would be useful for this insect or that insect. So cross application would possibly benefit from knowing the mechanism. What were you going to say?
Like it will help to predict the outcomes.
Yeah, predicting the outcome. So, for example, a lot of people are really interested in looking at different Wolbachia strains to see which one has the strongest virus suppression phenotype. And so there might be a scenario where you find this weird Wolbachia that has good, strong virus suppression, and you might wonder, well, does it have CI or not? It'd be real quick to just check to see if it has the genes and check to see if it would work. So predicting outcome of CI strength or compatibility with wild populations. That's another good point. What else? There's some other things too.
I guess for like our lab purposes, it addresses the argument between TA and 2x1.
Yes, but so I definitely think like I'm definitely interested in TA versus like 2x1. Like I'm interested in knowing. But sometimes I do kind of wonder, like, should we really be focusing on this? Is this like too nuanced? Or is it kind of like a waste of time? I'm kind of like walking through that thinking right now with you. Like all these things above suggest that it would be really important to know the mechanism. And also the TA versus 2x1, sort of like understanding that knowledge, that would be important for designing transgenic constructs. And people are now taking transgenic CI operons and putting them in the malaria mosquito, Anopheles gambiae, and they're inducing CI via transgenic constructs. So the best transgenic design is going to be the most simple. And so if you know which of these is the correct one, then it's going to help you sort of like make constructs a little bit better. I think that's a decent argument. Also, too, I think that the mechanism is super important for comparing techniques. And Wolbachia biocontrol is not the only biocontrol, right? There's other biocontrols which we have not talked about. There's precision-guided sterile insect technique, which uses CRISPR-Cas9. There are, now I'm like brain farting. Oh, there's Riddle, release of insects with a dominant lethal, which we might talk about. We'll talk about both these things. And these are all like promising technologies. And I don't think one is going to be the solution for everything. There might be, in this situation, you want to use this, or in this situation, you want to use this. And knowing the mechanism would allow you to be able to decide, well, which one of these is going to be the best for this particular situation. And also, too, when you're releasing insects, guess who decides whether you can do that or not? The people that live there and the government. And so for the government to actually make a good decision and for the people of that government to make a good decision, they really need to know the mechanism. So knowing the mechanism is going to be good for like comparing the techniques and making good decisions. So that's one argument for why, even though this paper is getting like very like into the details, these are some arguments for why this is like worth doing. Aside from just the basic, like we want to understand the biology. Okay, so let's just start looking at the figures. Okay, so what is this? What am I looking at? What's A? We'll dissect this one by one. What's A? What's happening in A?
Comparing two strains of Wolbachia.
Yes. Okay, so it's comparing two strains of Wolbachia. So here's wNo, here's wPIP. And both of these have a SYN type operon. So it's an operon that has this nuclease domain. And they're comparing both the A, the rescue factor. So in theory, this is the rescue factor. And in theory, this is the toxin, depending on what hypothesis is correct. What other things are they showing you here that are worth like pointing out?
Nuclease domains.
How many are there?
Two.
So there's two. So there's two nuclease domains. There's nuclease domain 1, nuclease domain 2. And so that's sort of like a staple in these SYNs is they have these two nuclease domains. The other thing to point out about this wPIP is there's another paper, you guys probably have not read it yet, but there's another paper where they describe the SYN wPIP system and they validate that it is a CI inducer. And so what you're not knowing is that context. And so what they're doing here in this figure is they're showing you this is the one that nobody's tested before, but it looks just like it's structurally similar to this other one that does induce CI. Does that make sense? Okay. What are these things, and why are they pointing this out?
Those are amino acids at certain positions.
Yes. So these are the catalytic triads. So these are the things that are actually like responsible for the DNA getting cut. This D, the E, and the K. Okay, so these are sort of like, of all the protein, these are really important residues. If these are messed up, it's not going to cut. And so they're showing you that they're in the active one, they're conserved, and in this wNo one, they're conserved. So the hypothesis would be that this wNo one is active. That would be sort of like the hypothesis. And then this is sort of just like a zoomed in, where they're doing an actual alignment of the amino acids. And here you see, here's the totally conserved across. These ones are the U means upstream. So that's the one on the left, 5'. And D means downstream, the one on the right, 3'. And so they're doing an alignment of both the upstream and the downstream amongst different strains of Wolbachia. And the residues DEK are conserved, and there's also this motif here that's conserved, and there's also some E's that are conserved. Essentially, they're saying, like, it looks like it's active. That's the conclusion. The conclusion is it looks active. Okay. Everybody get that?
What's WSTR again?
That is Leodelphax striatellus. I think it's a plant hopper, or I always get plant hoppers and leaf hoppers confused. Somebody fact check me. I think it's a plant hopper.
How come it doesn't, highlight the G there? For the
Wait, the G?
The E. Isn't that conserved in all the groups?
Where? What G?
It's all the way to the left.
All the way to the left. This?
The one that goes all.
Oh, why aren't they like putting a mark there?
Right.
It is all conserved, but it's probably, they probably don't know what that residue does, or perhaps they don't know if it's important for the actual structure. The ones they're marking are the ones they know for a fact are important in the biochemical activity.
Okay.
Does that make sense?
Yeah, it does.
But what you're suggesting is the fact that it is conserved, it probably does suggest that this residue is important for some reason. Maybe we don't know yet why. Okay. So that's that one. Okay, so now hopefully this was not too painful for people. Okay, this was rough. This is interesting. Okay, so let me first help you understand what this is, what to look for, and then I'll sort of show you what you're looking at, what you're seeing. What are these blocks? What are these white dots? They're yeast. Okay, and so what you're looking at here is really nuanced data. If you were to read some other papers, there's some other papers where the phenotypes are much more strong. So if you were looking for a big difference, unfortunately, there isn't a big difference in here, which is one reason why this data is a little bit difficult to interpret for a non-yeast expert, which is all the reason why I can help more. Okay, so it's definitely yeast. What do you think they're trying to show you?
Microbes.
It's growth. So that's all correct. You're looking at how well the yeast are growing or not, okay? And why are there 1, 2, 3, 4, 5, 6 different dot marks? It's a dilution. It's a serial dilution. Okay, so this is called a serial dilution. So you start with one concentration here, let's say 1x, and then usually these are 5-fold dilutions. So this concentration is 1/5 of that one, and then this one is 1/5 of that one, and then this one is 1/5 all the way down the line. Okay, so the reason that you do a serial dilution is because it allows you to see differences in growth. So essentially the speed of growth while you're expressing certain things. And this will allow you to see a phenotype. Okay. A couple more things about this before I sort of explain what to look at. What is EV? EV.
Seems like a non-viral.
What did you say? No virus is involved in this paper.
And then it said something about different alleles.
Empty vector.
It's the empty vector. So a vector is a plasmid. This is going to be an experiment where they're going to be expressing transgenes. And they're going to be putting them into a plasmid, which is called a vector. Okay? And then they're giving the yeast that quote-unquote vector. Why would you have a lane that's the empty vector? it's a negative control. So this is the negative control from which you're going to compare much of the things to. So if you're yeast and you just have like an empty plasmid, in theory that should not be a problem for you. Like you should grow perfectly normal. And so essentially like everything above this black line is perfectly normal yeast growth. This is like normal growth. Okay? Now what you want to look for when they start expressing these different things, anything that's growing less than the wild type, you're seeing some kind of a phenotype. You're seeing some kind of a phenotype. And remember that they're studying sort of like toxicity here. So they're studying if it's toxic, you should see a reduction in growth. Okay. Other things I need to define. Why is some of this galactose and some of this glucose? Did anybody catch that? Yeah, very good. So in these plasmids, you can clone a gene at the multi-cloning site, let's say gene X, and there's a promoter, and it's a gal promoter. So if you add galactose, the gene will turn on, okay? And if you add glucose, the gene will be off, okay? So now this is already a little bit easier to understand. What is this whole block then, where every yeast is growing really well? This is all entirely negative control. This is showing you that if you're not expressing any like weird genes, these yeast strains all grow perfectly fine because they're shut off with the glucose. Now this is sort of like, this is the stuff that they're testing when they're putting in different transgenes and they're turning them on and they want to see what the effect is. Okay. Okay, let me say a couple other things about this. The one thing that's really nice about yeast cereal dilutions is they are super sensitive. Like you can see and you can detect very, very weak phenotypes. And so that's a really good thing because it's a very sensitive experiment. But it might be a bad thing in a sense of if you're looking for this and you hope to see some giant massive effect and you think that like some massive effect makes it more meaningful, then you would not like this data. Okay, so now this, EV plus B. Let me ask another question. In the negative control, why do they have two empty vectors?
CI factor.
But why two? Why not just one?
So
Because there's two, there's like A and B.
Yeah, they wanna eventually test A in the presence of B, whatever like the combination is. And so there needs to actually be two vectors. So there's essentially like a vector 1 and there's a vector 2. And if you're interested, one of these is using a leucine selection and one of these is using a uracil selectable oxytrophic markers. So essentially, like there's two plasmids and that's why there's two, is because they're gonna eventually test, you're laughing, what?
it was one of something you said back then about leucine and uracil, and then you said something that was like completely foreign.
Oh, that's okay. Like I'm sometimes I will throw in more technical terminology for the people who know what I'm talking about. Like I know you guys know what oxytrophic marker is.
That was the
Yeah. No, I'm just saying sometimes I'll like, you don't need to know that for the test, but I'm also giving this for sort of like, I'll probably post this lecture online. And so like, I'm also trying to give context for everybody, if that makes sense. Okay. So now let's look at this, EV plus B. So this is sort of like the first experiment. This is really kind of like an important experiment for their hypothesis. What should they expect in this where they're expressing an empty vector plus a vector that has the B, the syn B? Induction of what? CI, true, but this is yeast, not an insect. So instead of CI, what might you see? What do you say?
Toxicity.
Yeah, you might see some toxicity. Like if it's a toxin, you hope that you see some sort of like toxic phenotype, which would result in the yeast growing less. Okay, so let me ask a question. Do you think that this one here is growing less than this one above it? Yes, but very slightly. And so this is why it's like the data here is very nuanced. You really only see the phenotype at this dilution, this dilution, this dilution. And then it kind of starts to get like, but you definitely see an effect. Like it's an effect. And if you read other literature on this where they're doing this in some of the other systems, like in the WPIP system, it's way stronger. It's literally like totally dead or not. So this is sort of like a weak phenotype inducer in yeast, this wNo operon. But at least it's showing something, and if it shows something, you can like do some experiments to test it. Okay, so then what are these? So these are, it looks like it's the same thing similar. It's an empty vector plus a B, and then there's this weird notation, K275A. What's that?
Like a point mutation, a change in the amino acid.
It's a point mutation, and guess what they're mutating? They're mutating these important residues, these Ks, these important residues that control the function, okay? So they're essentially like they're trying to shut off these nuclease domains. So if it's 275 and 629, 275, that's this one, and then 629. So they're testing both these important residues. And what happens when they change? So the lysine is going to be important for the activity. When they change it to an alanine, that's going to shut off the enzyme. And what happens in the yeast when they shut the enzyme off? It's no longer toxic. And so they're demonstrating that those domains are sort of like the business end of the protein. They're essential to the toxic activity. And then they test A. Here's an interesting experiment, and it's been performed many, many, many times with many different systems. If the 2x1 hypothesis was correct, and A was also like an additional toxin, you would expect to see some kind of an effect when you're expressing an A, and they don't. And neither has anybody ever in any other system. Nobody has ever detected a toxic activity from A. So that's what you see here. A plus B, this is another very important experiment, which is why I think they're marking it in red. Why is A plus B an important experiment?
The rescue.
That's in theory like the rescue experiment in the yeast. And so in this one, you can see that this is definitely growing better than this one up here. So you'd compare this to that, okay, and it's growing better. Although again, like the data is like super nuanced. Okay, now let me see what these are. Oh, so here they're testing, why do they do this? It's an A with these mutated Bs, but the Bs aren't giving you any effect. I think they're just like hammering in the idea that A is not a toxin. Like if A was a toxin, like maybe in these other combinations you'd see something. So essentially, like, what are the conclusions from this figure? Now that we kind of hopefully understand it, like, what are the conclusions? Alec.
It's not successfully revising it.
What do you mean?
It's not doing good.
What's not doing good?
I'm still lost.
You know the thing.
Okay, okay. What's one important? Let me just make sure you understand what's going on. What is one important take home from this data? Is there anything that you can look in here? You can see what they're doing, and you can say this is a conclusion from that.
Well, the A and B together on the far right is completely dying off
Over here?
on the glucose.
Well, that's a negative control. That's because they're not even expressing those genes. They should be growing. Everything should be perfectly growing on the glucose because nothing is being expressed.
Well, the A and EV have nothing.
A and EV. What do you mean?
I mean, the A plus empty vector just dies off over time, like faster than the others.
This is not dying off over time. These are
More toxicity then.
these are serial dilutions. So the fact that you see like 4 dots here does not mean that yeast is dying. That's a dilution.
This isn't like the time on the bottom axis sort of thing where it's
No. No, these are all grown for the same amount of time. It's just a different dilution. So the cells are plated at lighter density. What suggests that something is dying is if you compare, say, this to this, and this has less than this. That means it's dying.
What did you just move? Your mouse didn't move.
Oh, it's not marking it? Okay, so if you're gonna check, the point is to compare this to this.
Right.
Does this have the bottom one have less than the top one?
Right.
That means it's dying.
Yes.
Not the fact that this has less than this
Okay,
than the
see,
one
I was
over.
reading this completely wrong.
Yeah, okay. Well, now are you reading it correctly?
Yes.
Okay. Is everybody reading it correctly? Okay. So now what are the conclusions from the red dots? these experiments, the important ones. What are the take-homes? I am not going to tell you. You're going to have to tell me, or I will not move ahead.
All right, stay here till 8. All right, let's try number 1. It looks like they don't do as good as when they have that A.
Wait, what's number 1?
Like guess number 1.
Guess number 1. It looks like what does not do as well?
B plus the 2 red dots do not do as well as the ones above them.
This one? Really? You think this
I
one?
can't see what you're pointing at.
This red dot. You're saying this one does not do as well as that one. Really?
Those look pretty similar.
They look exactly the same.
Yeah.
So don't doubt yourself. Yeah, I mean, they look exactly the same. So why are you saying that this one looks like it's not doing well?
I'm saying that one and then the other one with the red dot do not look like they're doing as well compared to the 3 in the middle of them.
But you just said this one looks like it is doing as well as that one. Maybe start with this one. Start with this one. This one's easier. This is where you actually see something.
Yes. Well,
Okay, so
that one's definitely not doing as good as the one on top.
yes. Okay, so for people online, it was said that this line is definitely not doing as well as this one on top. Correct. I agree.
Okay.
So what is the conclusion from that?
That it has more toxicity.
What has more toxicity?
The B.
The B, yes. So now say it in a sentence.
B has more toxicity.
B is toxic.
B is toxic.
How about that? Does that make sense?
Fantastic.
Okay. I'm not picking on you. I really want you to understand that. That's all. Yeah, you're doing good. You got the right conclusion. Although whether there's a right or wrong conclusion, maybe that's a bad thought. But okay. Now try it with this one. Okay.
A is less, or B is less toxic in the presence of A.
Yes. Okay, that's good. That's a good way to say it. Very good. B is, let's say not toxic. Not toxic when you add A. Very good. And so what is this evidence for? Is this evidence for 2x1 or TA?
TA.
Yes. Good. That's the conclusion. Like this is supporting the TA model. Very good.
Why did they do 2 different temperatures?
So sometimes phenotypes, again, so the reason that they're doing multiple temperatures is this is a super weak phenotype, and you can exacerbate phenotypes by raising the temperature a little bit. So if they raise the temperature, they're able to measure it better. Does that make sense? That's essentially, that's why.
So I'm assuming they did multiple replicates, and this is maybe the best representation of what.
Typically, when you do a serial dilution, whenever they show you an image, it must, by requirement, be representative of at least 3 triplicate experiments,
Okay.
probably more. So whenever I publish a serial dilution, yeah, you'd probably do it like 6 times.
Uh-huh.
Hopefully, you get the same phenotype all 6, and then yeah, you publish sort of like the best image.
But you said the data kind of sucks in this one, right?
Well, so let me clarify that in a sense of, I don't want to say that the data sucks, because my point about the serial dilutions is they're super sensitive. So they're actually you could think about it totally the opposite way. You could think about that this experiment is so sensitive, it's able to detect a very weak phenotype. And the person who did this, their skill is high in a sense of they're able to do a good serial dilution where they can detect a weak phenotype. But the data sucks in a sense of you would hope to see something that's more blatantly obvious.
So they're not dumb, they're just looking for what's not normally looked at. It's like looking for something that's, you said, not as obvious.
Yeah, they're looking for something that's not as obvious
so this
in this
paper was
case.
published for a reason, and it's great.
Well, I don't know. You seem like you have. What are you suggesting?
No, I was just wondering.
This is a paper from my postdoctoral mentor. I am not shitting on the paper. Believe me. I'm saying, and actually, like this is not the coolest part of this paper. I'm saying this data is very nuanced, and if you're not a yeast expert, it's kind of. Wait, what? Maybe I should not post this discussion.
My bad.
No, but wait, did I answer your question?
Yes, it was a serious question.
And-
I didn't mean to get you in trouble.
No, it's not going to get me in trouble. Wait, but let me just make sure I did answer it. What was the question?
The original question
Yeah.
was at the beginning. I didn't know that like what you were saying is like it was looking at phenotypes that aren't as obvious. I thought you were
Yeah.
the data is not the best.
No, the data is very
So
good.
That's okay. That's
Yeah.
what I thought you said.
The data is good. So the person who did this, like I said, I'll reiterate just to clarify for everybody online. The person who did this is very technically talented in that they're able to see a very weak phenotype. But it kind of, in a sense of, like you're always, as a scientist, you always kind of hope for a blatantly obvious phenotype because it makes people feel better about your conclusions if it's like night and day. Does that make sense? Okay. So we're already like 40 minutes in. I'm gonna skip some of this other. I'll move to some of the other super important things. You want me to? No, I'll do the micro. I don't want to do the microscopy right now. Actually, okay. So what should I move to then? Since we are kind of running out of time, let's look at this. This is actually the most important figure. This is the most important. Okay, so what is this? It's their crosses. Okay, so what crosses of what insect? Let's help everybody out here. Wait, what insect? What bug are they working with? Is it a bug or is it like mice? It's Drosophila. Okay, so I would hope that we read the paper enough to know that it's Drosophila. Okay, so it is Drosophila, so that's what we're working with. Here's a male symbol, so they're crossing males to females. Okay, here's a wild-type cross. This is remember I told you last time how you measure CI. So the x-axis is embryo hatch rate, so this is a percentage, okay? This is 1, that's 100% hatching. If it's over here, that's a 0% hatching. They're all dead if it's over here, okay? So wild type, what is that? Why are they doing that cross?
It's a control.
Yeah, this is a positive control. Like how many Drosophila eggs should hatch if you just have a wild type mating to a wild type? And it's almost 100%. Typically, anything between 90 to 100% is pretty normal for Drosophila, for wild type hatch rates. Okay, so up here, everything's hatching. That's a normal, that's the positive control. Or the negative control for CI, like nothing's happening. Okay, then this one, what are they testing? An A in a male crossed to a female wild type. So what is that actually testing? If A is toxic. And is A toxic?
Slightly?
Is it? Because it says not significant.
Oh, okay, then no.
Definitely like not toxic at all. Like its mean is not even any different from the wild type. Does that make sense?
Yes.
Okay, good. So the conclusion here, what's your conclusion here from this?
A is not toxic.
A is not toxic. Okay, good. This is when you put the two together. So there's syn A, don't worry about the T2A. So it's syn A plus syn B. In a male, what's happening?
They still die.
They die. It's almost complete CI. It's like complete CI, okay? So if you have A plus B, that gives you complete CI. And then look at this. What's this? Just B by itself. And what do you see? Super toxic, okay? Maybe even a little bit more, but they're probably not significantly different. So B by itself, B is you could say toxic or it's giving you CI. So what's the big conclusion from this? It's sufficient. Very good. B is sufficient for CI. And so here's what I was talking about. Like, this is why I'm saying this is their best figure. Here you're actually seeing like blatant, obvious phenotypes. So response to Kaylee. Like here you're seeing like, this is a huge phenotype. It's not hard to see. Does that make sense?
Yeah.
That's what I meant by like, this is what you hope for as a scientist. Okay. Now these experiments, okay. So here are the two CI inducers, either A plus B or just B alone, crossed to an A. So what are these testing? These are testing rescue, and they're completely rescuing, okay? So the conclusion is A does rescue, A is sufficient for rescue, and B is sufficient for CI induction. So which one is correct, TA or 2x1? TA. In this system,
Let's say,
In-
is there still disagreement?
there is still disagreement. And one of the reasons that there could still be feasible normal disagreement is because there's no reason that they couldn't be different.
You can't look at all of the different.
You can't generalize.
types of organisms.
Yes.
Organisms.
And that's actually what they write in the discussion. They say that these data, I don't think that they say that the 2x1 is wrong. I think they say that these data argue against the generalization of the 2x1 model. So 2x1, I think, is kind of like a fluke of the WML system. These data argue that it doesn't apply to all systems. Let's see. Oh, this is just test. This is sort of like the main figure. We're already at 50 minutes, so we'll kind of like questions. Yeah,
Can you explain real quick the SYN A to TA SYN B system?
like what that is.
Yeah, that kind of threw me off a little bit when I was reading it.
Okay, so what's a T2A? Because I talked about that in biotech class. It's a virus peptide. So viruses often have long genomes that look like this. And sometimes those genomes code for multiple proteins. So the virus has to solve a conundrum. How do you get multiple protein from a single gene? Okay. And sometimes they've evolved this unique, interesting sequence called a 2A peptide. And if you put a 2A peptide sequence in your gene, when the ribosome reads this as messenger RNA, it skips. And so it'll code for a protein 1 here. And then over here, it would code this as a protein 2.
See, I understood that part, but I didn't understand how in the system of when they put it in, it didn't rescue it.
Yeah.
It didn't rescue immediately. Okay, that's a good question. So did you read their figure 5? That answers that question. So Kyle's asking the question, how come in the male, if you have A plus B, how do you even get CI? How does CI make sense if you have the rescue factor there all the time? That's a good question. I think that's what you're asking, right? How do you guys explain this?
Get some toxin escape.
Maybe.
A doesn't always bind to all of it.
The A degrades in the egg.
Yes. So this is what I covered before and if you look at their figure 5, they talk about this. The hypothesis is that in the sperm, Wolbachia are making, let's code A red and let's code B blue. They're making both, okay? So the modification might end up as both, but then what the thought is when this sperm fertilizes an egg, at some point something causes the A to degrade, and then it activates the B.
Oh, and if thus the egg is not producing the A, it will continue with the CI.
So one sort of like compromise between the 2x1 and the TA is that maybe one function of A is to prevent premature toxicity of B. You only want B, you don't want B killing sperm when they're in the male. That's too early. You want them to kill them right at the zygote stage. And so A might have a role in preventing that activity from happening until the right event happens. And anytime in cells, there's all kinds of signaling events. Like it could easily evolve a sensing mechanism to sense when is the right time, and then it would end up getting degraded, activate B, and then you get the phenotype.
That would be the 2A system on the opposite, correct?
The what?
That would be the 2A system on the opposite, because 2A system.
I don't know what you mean by the 2A system. That's just sort of a transgenic trick to get two proteins from one gene.
Or the 2x1, sorry, the 2x1. That'd be like the 2x1 system on the other end.
I don't know what you mean by 2x1 on the other end.
You need both for the toxicity, but in this instance, you need both for
For it to work properly.
yes. So like it'd be like 2x1, but in a different sense.
In a different sense. And that's kind of what I've always said to them. Like, no, I don't agree with your idea, the way that you're phrasing it. I could compromise in a sense of A might have some sort of like packaging role. Does that make sense? But these data argue that it's not necessary. These data argue, this data argues you do not need that.
How early did they go into embryonic development? Like, how early? I'm sorry. So are they dealing with just the gamete, or are they dealing with the I mean, they're just the zygote, or are they dealing with the gametes as well? Like,
Well, there's no direct like harvesting or analysis of sperm.
Right.
What they're doing is they're allowing the Drosophila to mate, they're laying an egg, and then they're looking at whether or not those eggs hatch. And you can tell, just to respond to your microscopy comment, there's a classical phenotype that you observe with CI, which is those chromosome bridges, like they're like strings that look like they're between chromosomes. That's an indication of improper mitotic division, and that's a sign of cytoplasmic incompatibility. Like you see that in wild cytoplasmic incompatibility. So the reason they did the microscopy is to show that when you have these transgenes, you're getting a phenotype that's similar to the wild Wolbachia. Does that make sense? Okay. Yeah. Dylan.
I might be remembering wrong, but didn't you say in the last lecture you tried to get a SIN B line and you couldn't do it?
SID B W pip. I tried to get SID B W pip alone in a fruit fly, and it would just straight up kill them. They were able to get a SIN B, so it's a different gene from wNo in Drosophila alone. And so that's why in the beginning I said one of the areas of low-hanging fruit after the initial sort of like characterization was there's all these other different orthologs with different sequences. Let's see how they work. Maybe we'll get a better sort of experimental system. And they did. They got a better experimental system with this version. Yeah. No, this is cool. I left CI, so although everybody else is like, I'm so.
The two TA system, could you?
What you mean 2x1 or you mean T2A?
2 TA, sorry.
Wait, there's no such thing as 2TA.
T2A.
What am I saying? 2 TA?
You're confusing T2A with 2x1.
No,
You can just say 2x1 cup to.
No, I know the difference. I think
Okay.
I'm saying 2 T said T2.
Yeah.
Okay, so what's your question?
So, in the instance that you could possibly check for the degradation of the SIN A, could you do the cross and
So okay.
harvest the eggs and then check like?
Yeah, okay, so wait. So what you bring up is a good, like, if you think about, like, what are the next steps? Okay, so you really would want to check for that degradation. How would you measure it?
You could harvest the eggs, both like,
Eggs or sperm?
probably the eggs. You do both. You could do both because then you could get the before, like your positive control showing is in.
Okay, so let's say you harvest some sperm and you harvest some eggs separately and you do some microscopy. What are you hoping to see?
Well, I wouldn't say microscopy. I was saying like you could check for the proteins.
How would you check for the proteins?
You just grind it up and do the extract, or like just do like an SDS on it.
So you're saying do an SDS page.
Yeah, do an
It would be
SDS.
way easier to do microscopy. Alec, help him out here. What could you do?
Microscopy?
Yeah.
Oh.
So what would you do?
I don't know, make like a plasmid that had GFP.
You would tag. You'd make some tag. I don't think they're using GFP. I think they're using immunofluorescence, so they're using antibodies. But you'd make some tagged A, and then you'd make some tagged, a different tag, like RFP or whatever, B, and then you would do the microscopy, and you'd simply ask, is A and B both there in the sperm, or is just one there? And I'm telling you that 2 competing labs have done those experiments and seen exactly opposite things.
So you're talking about like co-localizations.
Yeah.
Okay, interesting.
Yes. And so the one experiment where a group who I highly respect did it on they did these experiments not with GFP, this is just an example, but they have tagged A's and B's and they looked at it in QLEX, and what they saw is they saw that B was only present and observable in the late sperm and A was gone. And when they looked in the egg, it was both A and B.
They could try bimolecular complementation fluorescence tests where the two only glow when they interact with each other.
Well, we know that they interact. We already know that.
They know where.
Okay, so if you're looking at localization, they've already studied that. So what they notice is that when A is around, it pulls B out of the nucleus. I remember I mentioned that before. So anyway, the point is like there are people who are doing those experiments like as we speak. So the idea that you're saying these are the next steps, that's correct. These are the next steps. People are doing that, working on that right now to try to sort of like hammer in the coffin like which one of these is correct, if that makes sense. But there's a lot of debate. Okay, good. Are we done?
Yes.
Okay. Let me stop.
I'm a patriarch of science. Come to me for guidance. high, and I don't fucking lie. I'm a patriarch of science. Come to me for guidance. Come to me for guidance. Ooh, bitch, get out the way, get out the way, get out the way. Ooh, bitch, get out the way, get out the way, get out the way. I'm a patriarch of science. Come to me for guidance. Get out the way, get out the way, get out the way. Ooh, bitch, get out the way, get out the way, get out the way. Ooh, bitch, get out the way, get out the way, get out the way. Ooh, bitch, get out the way, get out the way, get out the way. I'm a man and the king, I wear the fucking ring. I discovered Sydney, so don't fuck with me. My fecundity is high, and I don't fucking lie. I'm a patriarch of science. Come to me for guidance. Come to me for guidance. Ooh, bitch, get out the way, get out the way, get out the way. Ooh, bitch, get out the way, get out the way, get out the way. I'm a patriarch of science. Come to me for guidance. Come to me for guidance. Come to me for guidance.